Hi, my name is Brittany Meyer, and this is my presentation on the research paper that I have chosen to study. The research paper that I have found is called Transgenerational Sterility of Piwi mutants represents a dynamic form of adult reproductive diapause. This paper was worked on by many researchers, one of which was Dr. Shani Maud. Dr. Shani Maud is a professor at the UNC School of Medicine, and he specializes in genetics and biology. The researchers looked at C. elegans, also known as roundworms. These roundworms undergo most biological processes but do not have a consciousness. The body of the worm is also transparent, which means that all cells can be visible with a microscope. These C. elegans were evaluated to see if they underwent heritable stress, and thus they experienced adult reproductive diapause. Adult reproductive diapause is the term used for when an organism becomes sterile due to stress caused by the environment around such organism. Sometimes the fertility can be restored in organisms, but not in all. To evaluate this, the researchers looked at the Piwi proteins found within the C. elegans. These proteins stabilize the piRNA found within testes. The specific protein that was tested was the PRG1 protein. These proteins are found in the germline, what allows genetic information to pass to the next generation. A method used to evaluate if these underwent heritable stress, and thus also through a form of adult reproductive diapause, was the visualization of germlines. Sterile worms were stained using a blue fluorescent DNA stain, also known as DAPI staining. This was conducted so that the germlines were able to be seen better through a microscope. The germlines were then grouped into 5 different categories based on comparisons to a wild-type control. The groups consisted of tumorous, large, short, atrophy, and empty. The tumorous, as the name suggests, resembled a tumor. The large germlines were the closest to the wild-type control since they had both meiotic and mitotic cells. The short were also similar to the control since they had meiotic and mitotic cells, but as the name would suggest, they were much shorter than the large germlines. Those that were characterized as atrophy had no meiotic cells, but they had uniformly mitotic germ cells. And last but not least, the empty germ cells had little to no germ cells found within them. This process was repeated, but this time the PRG1 mutants were crossed with CPLS36. The results from this were about the same as they had been in the initial categorization. The tumors exhibited more intense fluorescence, the large were about the same as the control, the short were shorter than the control, the atrophy had little fluorescence, and the empty had no neon green expression, which meant they were missing germ cells. The study found that they did undergo heritable stress and thus underwent adult reproductive diapause. Thank you.